My name is Mike Evans, part owner of Integrated Ag Solutions in Dodge City, Iowa, and teaming up with Clint and Luke at Hiawatha Irrigation Clinic. We'll get rolling here on the plant sap analysis and why we have moved towards that over the last couple of years. So, just to start off here, I've got a chart on sap analysis. It's on my right, your left, and the tissue. It really kind of goes through the differences of the two. One thing is sap is a liquid extraction. Basically, you take the leaf, they have a special pressure, they press it, and it squeezes out the xylem phloem sap, and that's what they measure. Tissue is a whole plant ash analysis, and that's where we get the numbers from. And the other side of it, that is a difference. That sap takes about 20 minutes between drying, weighing, and analyzing with the tissue. It takes about 12 hours, so it's a little bit speedier on the sap as well. And the last thing too is with the nitrogen piece, it's a little more sensitive to that, so that's why we pack and cool them and overnight them when we ship them. So here's just a visual, kind of give you an idea in your head what the tissue looks like typically. Leaves into an oven, ashed out, extracted. When we look at sap analysis, here's a cross-section of a leaf. Apparently somebody doesn't like my presentation. The xylem phloem inside the leaf, that's where the sap is pulled from. We don't use anything else in the leaf. We just pull that liquid out and send it to the lab and analyzes it. And why is this important? Probably the number one thing with sap is nutrient mobility in the plant, and that's what we're measuring. So the xylem is, think of it as a railway. It is from the root through the plant up. And that's taking water and minerals up. So that's the function of the xylem. And then the phloem on left here or right to you is more like a highway. It'll also pull nutrients up, but it also sends the exudates, proteins, sugars, the photosynthates in the plant back through the system to the dominant sink. So that's the mobility in the plant, illustrated by this diagram right here very well, but that's what we're measuring. And we get readings on that. So when we're talking nutrient mobility, there is immobile nutrients and mobile nutrients. And this diagram here illustrates the immobile calcium, manganese, boron, iron, copper, zinc, cobalt, and nickel are your immobile nutrients. And when they're deficient, they show up in the new growth of the plant. Mobile nutrients are what's left, so your nitrogen, phosphate, potassium, magnesium, chloride, sulfur. Sulfur is highlighted because sulfur will only move when nitrogen moves. So just keep that in mind. And then moly is also a mobile nutrient, but we just don't see it very much in the plant sap analysis, so it's a little. But these are what deficiencies show up in mobile nutrients in the bottom of the plant. So that's where you'll see these show up when you're out there walking fields. So here, excuse me, we asked the question, why paired samples are needed for sap analysis? And the nutrient mobility is the reason. Because we are taking a sample of the old growth and new growth, and we are comparing those two. So we can see those mobile nutrients and where they're flowing through the plant. And there's two distinctions here we need to make. Tissue sample is typically always new growth. So that's where we're pulling the sample from, new growth. Whereas the sap sample, we're recording old growth and new growth. So just keep that in mind when we're looking at the differences between the two. So on this slide, I kind of show typically in a tissue sample, this is what you'll get back on a report for as minerals, what we've seen historically. Some labs are a little different on their trace elements. But when we take a sap sample, we fill in all the blanks. We get 1 micros, and we get the nitrogen outside of the plant as well. We can look at to see functions and plant health as well through those. So it's a far greater scope of information coming back to us we need to fill in gaps. So here's the report, very large and a lot of information on it. It can be overwhelming. I know it was for me the first time I looked at this. A lot going on. We'll try to break it down, but there we go. Talking cation balance, kind of talked about that. Clint and Luke did with the rapid soil. Same thing applies in the plant, and we can kind of break those down. So here's the cations, positively charged ions. So this is what we can see on the report. And the mobile nutrients here are highlighted in the blue bar or red bar with a percentage. All the mobile nutrients will come with that on there. So we can see if we're excessive, following Wallace's law of maximum, or Liebig's law of the minimum, which is the whole barrel and stave. I think everybody's probably seen that quite a few times.
One thing to keep in mind when you look at the SAP report, they've got the numbers farthest to your right. Those are what we're actually testing. That is what was measured in this SAP report. The numbers to your left, those are what we call the operating levels, where the lab would like the levels to be, or we kind of refer to them as benchmarks.
Yep. Good point, Clint.
So it gives you an idea of where we want those ranges to be. So when we talk about balance within the cations or within the anions, what we're after is keeping those ratios in check between them. We want them to be close to the benchmarks or at them, but we don't, anytime one gets way above or way below, that's when we got problems.
Yep. Moving on, the other side that I personally like is what we call the nitrogen story. The advantage of the SAP sample in this part of the report is we do get total nitrogen in your tissues, but the thing that SAP brings to the game is ammonium, nitrate, and nitrogen conversion efficiency. So we can look at the two different forms of nitrogen the plant uptakes and how well it is converting them. So if we know if our plant's coming along very well or we got an issue going on.
This is basically a way to measure assimilation. When we talk about keeping them assimilated, in farming, I've always been talked about with nitrogen is how do we keep it, how do we stabilize it? But there's a lot bigger story to it. I mean, it goes to really the end result of nitrogen. We want it to go to amino acid and a protein. That's where it's usable. Darren earlier hit on this, that we can measure this in the plant. We can tell exactly if you're assimilating or not. This should happen in the root. We want assimilation to happen in the root. We want it to be processed to a protein. However, when we front load, because of logistics, we get these high dumps. So this is basically a guide of, hey, if we're not assimilating and we've got ammonium and nitrate in the plant, we got tools, whether it's carbon, micronutrients to help with the assimilation of it.
Yep, exactly. So plant sap analysis and the rapid soil, how they calibrate with each other, and they do calibrate. Here's the data set. I think this is your guys right out of Illinois.
Yeah.
And about 450 total samples taken over the course of the growing season. And you can explain this as your data set from it.
Yeah, so this is where we started. Luke and I started with tissue. It was a lot of valuable insights for us. Figured out nutrient limiting factors, and we was able to incorporate in-season nutrient management plans around it. So this is our entire data set from 2020, 2021, and then in between 21 and 22, a large chunk of our customers, we started side dressing potassium in our mix. And we typically side dress at V6 to V8, and you can see at the V10 how we bumped potassium. And this has been, for us locally, the addition to potassium in our side dress has been 9 to 15 bushel for us. So it really comes back to the balance, keeping nitrogen and potassium balanced. However, the bottom here is SAP. This is from last year. That red line, that is where our benchmark, or where the lab says we want to try to be with potassium. We do a lot with potassium management. We're foliaring a lot of foliar apps. We do side dress potassium. We break our fall base rate budget down, but we still mobilize. here in July and August, we mobilize out of the old growth of the plant to support the new growth of the plant or to support the kernel. So there's still room for improvement to take yield higher, but we're in the right trend with this.
So if you look at, this is last year's complete SAP database on the cations. So we have calcium, potassium, sodium, magnesium. So just kind of look at the potassium, stick on that trend. You'll see up here, the orange bars are the whole plants, but we get into the old growth, new growth chart with the lighter blue being the new growth and the darker one being old growth. The red line here is the rapid soil numbers. And rapid soil and the old growth are what you can calibrate to. And you can almost see it follows a pretty good trend line. As that rapid soil, K goes down, our old growth, K goes down because the plant is remobilizing that potassium in the plant because it cannot get supported from the soil itself. So it goes back to what Clint and Luke were talking about with those water and rapid soils and how potassium gets out competed. This is what the plant is dealing with during the season from that soil. Here's the anions. Clint can talk about this.
This is tissue test from 20 through 22. And that gray bar is the optimal range. And this is where maybe tissue test has misled us a little bit. We thought we was always adequate in phosphorus application. So we, as a whole, had our customer base pull back phos in a big way. We focused those dollars elsewhere. And we thought we was doing okay, but phos is still a very limiting factor. As soon as we moved to SAP, rapid soil, we realized that phos as a whole is almost as big of a limiting factor as potassium. So what we really struggle with is the mobilization. We mobilize harder. We keep our new growth in check, but we really pull from the old growth to support. We're not getting enough flow from our soil. So how we get better in the future, I think we complex our applied phos. We shift some of those dollars to in season to bump that later in the season when there's a big phos demand in reproductive stages. And really what we're after with this is from a grower or farmer standpoint, how do you take this data and get better? The better job we do at keeping old and new growth together, keeping them very much close in capacity, and kicking that later in the season and not mobilizing that phos from lower in the plant to support the upper growth or to support the kernel or pod, it's going to equate to yield. Like some of the farmers we work with, it's made just an 8% difference year over year was big bushels.
Thanks. So here's the comprehensive data set on those anions, chloride, phos, sulfur, nitrate. We'll kind of jump into the phosphorus. And again, kind of highlighting the same as the potassium chart, that red line's the rapid soil phos number, and it almost just mimics the old growth here. As you follow that red line, the old growth chart just is right along with it. So the soil's not supplying it. Plants having to remobilize it from the bottom up to support the new growth or the reproductive portion of the plant. So it's pretty powerful information that we can pull together and make better management decisions as we grow the crop through the year. Now here's a chart, kind of in a different format with the cations, but we're kind of got these red squares here to show nutrient drops. If you look at that gray box, it kind of talks about drop ones around herbicide time, like V4, V6. You spray chemicals, will put a nutrient demand or slow the plant down, potentially, what Clint was talking about towards the end of his presentation. And then there's a couple other ones through the season, potassium later on in the year. But in this data set, 50% of the samples were below sufficiency levels, so below that lower operating limit. We can kind of show it there. On the anions, phos, chloride, sulfur, nitrate, same thing. If you look at phosphorus alone, you can see those critical drops early on in the season, kind of through that late vegetative and late season. And you can see during this hard push at the end of the season, we get a big draw as the plant's going in to fill in the seeds and getting those seeds ready to go. 74% of the phosphorus is deficient and 85% of the sulfur is deficient in that data set. So there's huge demand for those products. Here's sulfur. One unique thing about sulfur, it tends to be a little bit more early on in the season. There's more demand at times. And
when it comes to sulfur management, what we've found, it's so mobile in the soil and can be flushed and combined with lots of nutrients. We're very much into a slow drip of sulfur approach. We'll maybe use some with our base rate fertilizer plants, but then we use a lot of melted AMS 8009 through the season. ATS is a great product on the side dress, but melted AMS, a couple gallon in the season will completely move the sap needle. And that's one thing that's so cool about sap. We can calibrate our products to them. So for us internally, as we try to get better with formulation and products, we use sap very much as a guide. We can take 3 phosphorus products or say 3 sulfur products, spray them out, we'll pull a sap test, spray them out, come back 6 days later and see what the movement was in plant sap, how much that was uptake of each individual product. So as a company, we've kicked a lot of products out and brought a lot of products in based on being able to move this and then ultimately get yield out of it.
Yep. Here's the micros. It's we just highlighted a few just to show the variance. One neat thing, like the zinc is down here in the lower right. And you saw in the soil, it spiked here, and then we saw it in the plant a little later on. So one thing you'll notice in these charts, there's no red bar showing you the optimal level on there because most of these aren't hitting them. There's a reason why. We're just so deficient in soil and in the sap or in the leaf sap analysis that they haven't registered. So here's this chart again with critical drops. Probably the biggest thing out of our data set today that we've pulled is 100% of the samples are below zinc optimal levels. So it means all of them are deficient. Boron is 90%, iron 87%, and copper is 97%. So our micros are well below where they need to be based on the lab's numbers. So really opens your eye out to the micros and the needs for them.
The big thing we're finding with micronutrients, they create macronutrient efficiency. The better job we do if we allocate some dollars to micronutrients, we create better plant balance, which then allows us, we've probably got to go back and reevaluate NPK a little bit because they become more efficient in how they work within the plant.
So here's zinc, and that, just like Clint said, that relationship. Here's phos in the earlier slide when we saw that July 30th, really deficient mark. Look where zinc is at that same point in time. It's really low. So there's a relationship there, definitely showing up in the sap.
Zinc is the regulator for phos. It is what controls phos from flooding into the plant too fast or when it's too slow. So when zinc is at a low capacity, the plant has a very tough time regulating its phos intake. So a lot of times we've found where if we have a really deficient phos plant and we go out and apply a phos, but if zinc's at the bottom of the barrel, we're not going to move that phos into the plant very efficiently. We need a little bit of zinc in the mix as well.
Yep. So I kind of highlighted zinc. It's a worldwide deficiency. It's not an unknown problem to the industry. Probably why, if you go to retailer or anybody else, they talk about zinc and the micros, it's quite common. There's a lot of negatives to a zinc deficiency. Impairs plant defense, stress mitigation, creates leaky cells. To Clint's point, doesn't help with phos regulation as well if you don't have enough. But going back to what Clint said about calibrating products, here's a slide that we did a test on, our Flomax zinc. You can see these are pre-application, applied a cord here, then came back a couple days later, took a sap sample, 9 days later took another one, and then 16 days later took another sap. So we were able to watch that zinc in the plant and how long it held and moved through the plant. So here we can say this the zinc at a quart can last about 10 to 14 days. Then we'd have to reevaluate what's going on. So this is calibrating products to the sap. It really helps us understand what products are doing.
One thing I might add, when you spray a foliar nutritional product on your crop and it's straight nutritional, you're going to get a 10 to 14 day bump in the plant. So really it's all about how do we time, if we've got a really dry period and potassium's really weak in the plant, if we see rain coming, we can buy ourselves 10 to 13 days at a higher level and get yield out of it. However, when we start stacking carbons, we stack amino acids, plant extracts, we'll get a higher response period past 14 days. So that's the art of what we're trying to do, is how do we use the complexing agents, biostimulants with our applied nutrition to drive the process longer and create yield.
Here's copper. Looking at it, you can kind of see where those drops are or where it struggles. Copper's important in lignin, cell walls. I like to call it, or it's been called, books I've read called the elastic mineral. So think of it as rhododendron, as corn grows. Green snap is probably my best example. Copper helps defend that because of its elasticity and structure. Important in chlorophyll, enzyme, and defense as well. And we highlight that it's really key in nitrogen conversion. Same thing here, took a 4 max copper, applied a quart, and almost mimics what we saw in zinc as far as staying power, just in applying that product by itself. So just another example. Any questions on that?
Do you have a plan for how we address our micronutrient shortfalls?
Repeat the question. So what's the plan for addressing micronutrient shortfalls?
It's two-part. We can't apply enough micronutrition in liquid form to support the plant. We get a 14, 15-day bump max out of it. So I think in the future, we're going to build it into base rate. We really like some of the Midwestern BioAg dry fertilizer micronutrients. They're homogenized. They all have the same amount of micro in each prill. We're having really good luck with that. It's more cost effective to build it into your program. And then we use the liquids in season to critique. So we've got products that we're formulating that has our main 4 dysfunctionals: iron, copper, boron, and zinc. We also have a little manganese in there to make it easy in season, but it's got to be a 2-part, and micronutrients are immobile. So our plan is, as a whole, we'll maybe do a base rate fertility with dry micros, but then if a farmer's got $10 to spend on liquid micronutrients throughout the season, we're going to divide that up across his current passes. Because they're so consumptive in the plant, they get used so much, we found a lot better ROI instead of just hitting it one time, if that makes sense. So, great question.
So we're going to jump into nitrogen assimilation and kind of go over this. Back to this chart. Looking at the values here, you can see they're highlighted. This ammonia is 243, nitrates 381. But typically I look at first thing is the conversion efficiency. Like this one's 85%, this other one's 97 on the low leaf. Lab wants you at 95% minimum for assimilation or conversion. So anytime we're below that, there's a dysfunction going on within the plant for whatever reason, eliminating that, and we're not converting nitrogen. So here's basically just a diagram of the nitrogen cycle in the plant. There's nitrates, NO3, assimilates into NO2 to NH4, goes to the GO-GAP cycle to amino acids to protein. And then here's ammonia, goes that route. Anytime this gets interrupted and compromised, and like Clint said, if it's not done at the root tip, it gets put on the plant, it puts energy demand on the plant.
Darren did a great job hitting on this. And we can actually measure this with sap. That's the big thing I like, because ammonium flood typically comes early. As our organic soils warm up and our organic matter becomes active with biology, we get this huge flush of ammonium to the plant. It's dysfunctional, but part of that comes from front loading. Nitrate is predominantly the problem the rest of the season, it wants to come high. So the goal is to hold them at 50 ppms or less. And so like with specialty crops, there's a huge energy demand. So the secondary metabolites needed, the carbohydrates needed, they've got to be robbed from ATP photosynthesis to complete this process. It takes a huge amount of energy. But the thing we have found is there's a ton of micronutrients involved in this process. So if we apply some soluble carbon, say backbone, with iron, we'll drive ammonium out of the plant. Just boom, within 6 days. 16 ounces of, say, a Floam Max iron. But the same way with nitrate. When we get nitrate that's dysfunctional, we'll apply copper with soluble carbon, drive it out of the plant.
Yeah. And this here just kind of explains what Clint's talking about. But there's two main assimilation systems: nitrogen reduction and ammonia assimilation. When one fails, it stagnates the whole system, and we call that bottlenecking. And probably the biggest one we worry about, ammonia is concerned, but nitrate, if nitrate isn't converted, it gets stored in the vacuole for later processing, and that becomes an issue. As we store more nitrate, the plant gets, the vacuole gets bigger, the cell gets longer, thinner, creates more susceptibility to disease and insect attack, because that weakens the structure of the plant. And also, with the plant, go ahead.
One reason that is, nitrate causes extreme growth. When you get this excess nitrate, you get the vacuole of the cell gets bigger. It has more water internally in it, more moisture. And so when you get say a pathogenic disease that comes, a spore that lands on that leaf, it's trying to penetrate that leaf to find water. The bigger the cell, the more dysfunction nitrogen is. You got these big cells that they can easily penetrate. The disease becomes systemic, goes throughout the plant. When our plant's balanced and we're processing nitrogen, the cells become much, much tighter. When we have the calcium, the boron, the potassium, we get really tight cells. It becomes really hard for that disease or pathogen spore to find water in that situation. So that's the gist of what we're trying to get at. A lot of our high disease problems that Luke and I face back in Illinois come from guys that are planting 240 pounds a in. Those are the areas that we struggle with disease first before our fields that are more nutrient balanced.
And this highlighted here piece, I think is always interesting. 16% of the plant's total photosynthetic energy is used just to convert nitrate. So there's a higher energy demand on the plant as well if it has to be processed. Well, I think it is 1 molecule of nitrate is 3 water molecules. You take it, right?
Our guys we work with in Illinois and Indiana with vegetables, when they see dysfunctional N, airplane goes up because the quality is affected at the end.
So the essential minerals drive the process forward. Kind of highlighted them. Well, big thing is if carbohydrates or carbon isn't in this process adequate amounts, then it'll get shoved in the vacuole. If we don't have that first, that's the first thing that'll happen. The plant will just move it on because it cannot process it. Then the micronutrients and sulfur come into play. Like we see here, moly is important in the nitrate reductase enzyme that starts that process. If you have not enough moly, that process is stalled out and you get shoved in the vacuole. Copper, iron, zinc plays a role. Then phosphate is here at the bottom. So that's important to understand that when we start cycling nitrogen in the plant, we automatically put a demand on phosphate. Because 1 molecule of nitrate takes 16 molecules of phosphate to complete that cycle.
One thing, to his point, a lot of products coming out, biologicals, they create N2 fixation from the air. And where that takes place is on the little hairs of the leaf. They call them trichomes. When you activate a trichome to grab N2 from the air and do that is a very energy intensive process. A lot of companies out there making a lot of claims about how many pounds you can reduce when using products like that. And what we've found through our trials and a lot of our research, you activate N2 fixation from the air, you're going to put a 25% demand on phosphorus, iron, copper, zinc, just right off the bat. That is a very energy intensive process. So to Mike's point, when this end gets dysfunctional, it puts the demand on the phosphorus.
Yep. So that's with the carbon piece, we kind of hit on it earlier in the other presentation, the product backbone kind of helps fill that demand. One part I like to highlight is what we call the carbon skeletons. Those are just the compounds and structures that fill that gap up in there. And this kind of explains it here to the point of here is the whole cycle chart of it. Here's the carbon skeletons that are needed to fill out this pathway to protein. And if we're missing the carbon pieces in here, it does not move forward.
Question.
Yes?
So on the previous slide, you're talking about some of these biologicals that are creating nitrogen. And how that puts an added pressure on the other nutrients because it's so energy intensive. Would that explain why there's such a wide range of results from those products? Where if they were in a nutrient surplus of some other areas, maybe they actually do get that benefit, but we can't replicate it consistently.
100%.
I mean, that's been my frustration with those products,
Yep.
those claims, is because I'm not saying they're lying, but we haven't been able to replicate it consistently.
Yeah, we have to be able to measure it. And so part of Luke and I's past business, within BioAg, our first company we started, we was a biological research company. So we got paid by all sorts of companies, all the ones you see in magazines, to do research. And we took it to the field and did weekly sap, weekly tissue on them. And that's the kind of things we were seeing. Just that overall demand. So if you've got a great growing season or you've got high fertility under your soil, you got enough to support it. Or if you got adequate soil moisture, you usually have good nutrient release. But anytime there's stress, the plant doesn't have enough energy to complete all the processes.
And the problem is that a lot of the farmers who are intrigued by those kind of products tend to be minimalist in their applications and their fertility programs prior, and so it's just compounding the issue.
100%. So as a company, as BioAg, when we originally started, I went down that road. I went down to the road of no applied PK, very minimalist, 4/10 to half pound of nitrogen. And with our heavy soils where we're at in West Central Illinois, that worked great. However, we've got a lot of highly eroded soils. And what we found was, through our data, we was constantly doing dry fertilizer strips, fertility strips on our eroded soils. After about 3 or 4 years, yield started to go down in a hurry. So we've seen it. We've had to pivot as a company. And our new motto at BioAg is nutrient focused, pathway infused. Can they create efficiencies? Can these biostimulants and biology create efficiencies? Absolutely, but we can't ignore the minerals. We cannot ignore fertility. So, great question.
So kind of go over why backbone is a little different. We get this question quite a bit on humics, fulvics. We get a lot of information on there. Kind of basics on humic acid, large carbon molecule, really works best in the soil. Helps create a better CEC, all those things. And we've used a lot of humics, at least on Kelly's farm. I'm sure Clint, we've used a lot of them in the past. Fulvic acids, smaller carbon molecule, probably the great thing about them, they work across all pH ranges, and also they can carry and deliver 10 times their weight worth of nutrients through the plant cell membrane wall. So we like to call them a fullback essentially. They'll drag anything with it, not even minerals, but your herbicides tend to work well with them because they're latch onto that and pull along with it. Probably the biggest thing, I think Clint hit on this earlier, what backbone is, more of a diversity of carbon sources blended into a single source, organic, carboxylates, fulvic acid diversity, and just like I said earlier, it's biologically and enzymatically processed, so that gives it a little bit more, right?
Yeah, you retain your oxygen molecules, you retain your secondary metabolites when you have that process. So there's 3 main ways you get humic and fulvic fractions when you manufacture it. We use, in the industry, uses potassium hydroxide and low heat to extract humic and fulvic. It's pretty fast and usually retain quite a bit of the horsepower. However, you have a high pH. You have some mixability issues with acid fertilizers and different things. Then you've got your enzyme and biologically derived humics and fulvics. They take a long time. They're usually a little more expensive, usually have a more neutral pH. The third form that probably gives humics and fulvics a bad name is a nitric acid extraction fraction portion. So they tend to have a low pH. They're really quick to make. They extract all the humic and fulvic out really quickly, but you lose your oxygen molecule, you lose a lot of your secondary metabolites in the process. However, the potassium hydroxide, you know, so if we're just looking to stabilize urea or UAN, a lot of the potassium hydroxide extraction humics for a cheaper source will do the job all day long.
So here's kind of where we come up with a program. So on the left is kind of like Clint, has for their company over there, BioAg, their kind of base program on their site address. But kind of how we handle this stuff, you know, really testing, water, rapid plant sap analysis, that is key of everything we do. Getting the information and the data in to know what we need to do. And interpret that with those results, deploy a plan or a solution to help remediate any dysfunctions that we see, and then analyze it on the back end, validate it, and reassess. So that's kind of the process we take with Calibrated and helping growers and others. This is from Clint and Luke out in Illinois, just kind of an example of management with SAP versus not, and some of the results. And Clint can kind of hit on.
They're both of this field's good corn, but the big thing I want to challenge you guys on is think about mineral nutrition as a mode of action. When a plant is balanced nutritionally, we have the ability. The plant has many operating systems. It's got a defense system, what they call a systematic acquired resistance. It's also got a growth system. It's got all different sorts of systems. It can communicate with its environment, but the more nutritionally balanced we are, the better the plant can communicate and tolerate stress. So a lot of what we find is just when we bring balance in season with nutrition, we create a plant that doesn't get disease in it until later in the season. We don't see responses to multiple pass fungicide, and I think it's important to realize there's things that you can cut when you focus on nutrition.
This is just a trial we did. You can explain it, it's your data.
Yeah, so this is just a Y drop. So we do a lot of dry-based fertility studies. We've still got them ongoing. But a lot of times what we found is year over year, it's hard to get base rate fertility to pencil out. If you truly try to ROI base rate fertility in 1 year, it is very much a slow drip. When you apply fertilizer in the fall, very little is available to your crop. But it's a repeat deal to get dry fertilizer to pay. But if we've got to weather a tough economic storm, or if we've got problems on our farm financially, I think there's a great place we can weather multiple years without base rate fertility, go to some in-season nutrition apps. But this is just showing you the difference where we had no dry fertilizer compared to the side dress. And the NADE photos on this, or the NDVI photos, were really awesome. We seen the dry fertilizer strip in May. We side dressed in late May. We could see the Y drop strips where we did in late July. So great yield gains with this. However, our current system today, we are applying about 60% of University of Illinois, Iowa State fertility recs, and then we're coming back and using in-season management to fill it out. So.
From biology, any questions on all that before we dive into this subject? No? Perfect. Biology is a mode of action, a season-long approach. So it's a little different thinking with biology. Comes from Clint and Luke and Jared as us as a collective. But I always start with this because the soil food web, I think it's kind of important to show what this all incorporates in your soil. Actually, you know, we talk a lot about bacteria and fungi, but there's a whole community beyond that is integral in how that thing functions. And I think we believe in diversity and numbers as we talk to people. Those are just as important as anything in handling the season-long growth of a plant or soil. And I think from our experience, at least with Kelly and the Tree Mag, we've had a lot of biologicals come through the door and try them out. But they've been pretty standard biologicals and pretty well in this bacterial world. Maybe you get some fungi here and there. And talking about BioID, what Clint and Luke have brought to the table, you get a lot more diversity, filling in holes that could be created through the season. And I like this comment: balanced soil ecology equals lower dependence on inputs.
Yeah, and you know, that's the whole thing with biology. We're at a place now where being able to test our soils from a biological standpoint is here, and it's cost effective. I think that's where we're at. We've made, biology has gotten a very bad outlook amongst farmers because of its wide variance in performance. And so today, now we can actually measure what our weak links are. We can measure biofertility weak links. We can measure our pathogen load. And that's where we're at as a group at Calibrated. We want to be able to measure it, implement a product or program, and then measure on the backside to make sure we're actually getting it. So I don't like the word soil health. I think it's too broad. I think you have to be able to measure change, and that's what we're all about. So the law of competitive exclusion and the disease triangle. So when I talked about mineral nutrition as a mode of action against plant stress or plant disease stress, if we can eliminate the host through better mineral nutrition, the pathogen can't compete. And we have a balanced plant that the plant can communicate to stop diseases. One thing we've learned through our pathogen testing, the environment is pivotal. So we all get those times where rain won't stop, our environment changes, we go anaerobic, disease blows up. Those are challenges we can't often overcome, but like in our area, one thing we've noticed through the pathogen testing in the soil, we're in a very wet environment. And so where we pattern tile fields, our disease load in our soil goes down by 50%, just off doing a better job with surface drainage and subsurface drainage. But the more we can affect the host, the plant, with better nutrition and changing the soil environment, we're going to eliminate the pathogen. And the law of competitive exclusion just states when there's two species, so if you've got a pathogen and you've got a beneficial that controls the pathogen naturally, our soil ecosystem, our plants, they're very much can take care of themselves, but we become out of balance. And part of that is unintentional. Fungicides as a whole are non-selective. They kill the pathogen right now, but they kill the controlling partner of the pathogen. So when we quit using fungicides, a lot of times our disease goes up even higher because we don't have the control agent. But back to the law of competitive exclusion, when two species are competing for the same resource, whichever one has the higher number wins. And we'll show that in the next slide, I believe, Mike, won't we?
Yeah, maybe make it quicker here. So this slide here kind of emulate what Clint was talking about. Basically, these are two species that are competing for the same resource. Put it in a petri dish, timed out over time, and CFU counts as we went over time. So it's basically a 2-week span. But when they put them together in the same petri dish, do the same thing. One outcompetes the other one. So it's just a visual of what Clint was talking about. Pretty cool illustration.
So yeah, we've partnered with Earth Optics, Pattern Ag. They pull your standard base rate fertility test, but they also pull your overall diversity. They look at the overall biological diversity of your soil. They look at your pathogen load, as well as some of your biofertility pathways, how good we do at phosphorus optimization, from converting phos from unavailable forms to available forms and delivering it to the plant, how well we do on nitrogen. It gives us a starting place to say, where's my weak link in my soil and what product will work to control it. And we are a big believer in doing that so we can actually get to the measurement of all this and not really guess at what product's going to work. So on this next study, this is in Luke and I's backyards. This is split fields, or fields right adjacent to each other, same crop, same hybrid, same fertility program, same farmer. And then we just basically look at the pathogen load after we implemented our biological. So at the top, you've got your untreated, and this is data out of Earth Optics or Pattern. But these are diseases across the top. You got Fusarium, Pythium, and if you see a red bar, one thing we really like about pattern earth optics, it's very visual. It's very easy for anybody to understand after you get walked through it once or twice. But the red bars mean, hey, this is going to be a problem to the crop. This is going to eliminate, this is going to take yield off the table. The ones that are in orange or yellow color there, they're in between. They could be a problem and reduce yield kind of on the edge, but when we get down to the green, that is when we don't have really a problem. It's below CFU thresholds to be plant infectious. So CFUs is what they call colony forming units. That's how we measure biology in the soil or in products. And so this is just treated versus untreated in that pathogen profile. So BioAg as a whole as a product, it's very much a biocontrol agent, has beneficials that will attack pathogens. It's very much focused on biofertility, stress adaptation. It's a very diverse product. It's bacteria, fungi, protozoa, bacteriophages, beneficial viruses. It's a lot going on. It becomes a challenge to explain. But one point I do want to make, this is not a biofungicide. This is just data of how when biology is working for you, you get this natural reduction. And this is what we want. When we talk about, we get tired of fighting symptoms and we want to get to the dysfunction, this is exactly what we're talking about. We fight the symptom of disease, but the dysfunction is we don't have the control agent in our soil. And so we want to rebuild those populations back.
Here's just a sample of a soybean field with the same principles that Clint was laying out there. And then this one's, I like this one. This was a couple years ago what Clint did. Same kind of concept, long-term biology with BioAg put on the fields, and these are the untreated fields just on Goss's wilt. So it's very powerful data that helps us look at those things. And you can explain this slide a little bit.
Yeah, so this BioAg, we have 1,000+ species. We believe in the diversity. We don't feel today there's enough information to go single species strands as from product solutions. We want to build that diversity back in the soil. And we've had a great amount of success. We've really grown into specialty crops out west. And the one story I want to tell you guys, I'm worried like in our area, at our spot, we're now to the point where guys are going back to in-furrow fungicides, multiple 2 and 3 passes through the season. But as Luke and I travel around the country and we go down to pecan growers or pistachio growers in California, they have worked themselves into a problem with resistance to fungicide, with disease resistance to the over-application of fungicide. A lot of the fruits, nuts, and vegetables we eat are sprayed 7 to 13, 15 times a year with fungicide. And something I didn't realize until we started traveling around, but now they're going back to mineral nutrition as a mode of action, using plant sap to bring better nutritional balance and the biology to it. So these are some pictures from Idaho, very visual out there. Their soils, because of the intensive agriculture and growing vegetables, we see that. So more of a visual. Here in the Corn Belt where we've got pretty good soils, we don't usually see the visual like we see in that picture, but this is some of our plots. Building a biological program we feel is important. We use the biology. We usually have starter or micronutrients, and we usually have a carbon source loaded with these primary and secondary metabolites in there as a food source to activate the biology. The one thing we think about when we go ahead, Mike. When we think building a program, this is kind of our standard in-furrow program. We run starter, but we don't run very much. We want biology to do most of the work in creating those nutrient reactions and cycling nutrients to the plant. Aminocarb is our food source. Hydrogen Co is a hydronium acid, and we're lucky we get to work with some great microbiologists at universities that will take our products and actually look how, when we put this collection of this blend together, how it creates a synergy to the plant and actually gets the microbes to activate the rhizophagy process. So the rhizophagy process is where microbes actually carry nutrients and they enter through the root tip, and then they'll release those nutrients into the root system, and then those microbes will exit through the root hair. And that's what creates our root hairs and our root systems actually grow. It's that microbial interaction. But a lot of our success as a company has been figuring out how to put the right programs and products together to drive that synergistic approach in creating a program. High fertility in furrow corn program. So we got a lot of manure base. We change it up. We won't necessarily run starter fertilizer. We'll run micros and calcium. One of the dysfunctions we see in high fertility ground, especially manure ground, is we see this huge flow of chloride and sodium to the root in our early sap samples. And it completely outcompetes our phosphorus, sulfur, potassium, and calcium, that sodium and chloride load. One thing we've learned is when we have plant available calcium at the root tip, it actually will turn on anti-transport pathways for chloride and sodium. It's something we've been able to measure through the sap. Going back to, I would much rather a farmer, one of the things we've found with biology, it's a season-long approach. We've got broadcast programs, but I really like the 8 ounces all season long in every pass. Put a little backbone is there as a diverse carbon source to activate the biology, but it's also there to buffer it into the tank bed, buffer it with your fertility, protect it during that application, as well as with applied fertility. But we've had our greatest success in inflicting change and nutrient use efficiency, or in fighting these pathogens with a little bit in every pass. So there's a lot of claims from biology that you can reduce fertility X amount, or 50%, 40 pounds. You guys have all heard them. I don't really like that, because depending on our environment, if we're dry or if we're wet, that changes how biology works in the soil. It changes the efficiency of when you use biology. So we have always went to a validation of when we want to see what biology does as far as creating nutrient use efficiencies. This first chart to the left here, that is tissue test, control versus treated. Biology's on one side. This is a large data set with lots of different fields averaged together, but we do most of our work on phosphorus, potassium, calcium, micros. We affect all nutrients, not just one. Nitrogen's on the lower end of what we affect, but however, as we've moved to sap, we have done the same thing, taken a lot of samples, treated versus untreated, using biology and fertility blends, and then we measure the performance. When we go out west and we run this through pivots, we'll maybe run 100 pivots with it, 100 pivots without, and we'll see the P and K flow difference with just 8 ounces in there. But we help deliver the minerals. So as a company, we're very much focused on creating that synergy and helping deliver it, not just as a standalone. You don't need the mineral nutrition. This is all diseases decreased over time, over a 3-year period from using BioAg. We're happy to share this with you. And again, we're not a biofungicide, but this is just the data that we have found as we implement this program. So when you think about biology, ask a company, will they supply you with a report showing what's in their product from a microbial perspective, and how are they measuring at the field level to what the change is. So
that's it. Any questions?
Tying it back to irrigation water, do you have a sense of how, good or bad quality irrigation water, how is that going to affect all of those biological markers and presence?
This really comes back to great question. It comes back to Jared. So Luke and I spent a lot of time looking at different city water, different water treatments and how it affects biology. But in irrigation water, and especially putting it with acid fertilizer, 2 years ago I would have told you can't do it. However, as we've grown and are going in acid fertilizers, waters all the time, we see that P and K performance increase in the sap. So I think it's a full sin. I think the secret to it it's your mixing order. It's when you put your carbon in, you put your biology after. You buffer your biology with the carbon for that application ride, and you're pretty safe. They're pretty hardy as well. We found they're pretty hardy. So.
Guys, you covered a lot of ground today in 2 different presentations. And first step, and what's realistic the first year? Just give us a sense of what you're thinking. I know that we don't have this fully baked yet, but I guess it's an opportunity for us to share what do you think is our best first step and first year expectations. We're not going to light the world on fire the first year, but what does that look like in you guys' mind and your vision?
My vision is collecting that data. Being able to make the right decisions with collecting the right data, I think is paramount. I think the long term, we've got to take that base rate fertility and build it into what we're going to add in with the irrigation water and making it better. But I would encourage you to take a field and push the envelope. If you've already spent some base rate fertility, let's take a couple fields, push this acid in and see what we can affect as far as taking yield to the next level. Most of the pilot projects and projects we've had going, if a guy did one pivot or a little bit of drip, he's adding 3 and 4 the next year and adding to it. So as a business owner, I always feel good about that when guys are wanting to add to what we're doing. So I don't know if you got anything else to share, Mike.
Yeah, I just take what we did at Kelly's. I mean, it was, once we figured this out, it was, let's find the 2 or 3 things that are our biggest pain points. You know, like for Kelly with his water and things we do, I mean, sodium's high, potassium's in the tank. So how do we fix that? And that's not a flip overnight either. We've got to mess with a few things and then we got to apply it to the field. So I think that's the biggest thing, is trying to figure out the 1 or 2 things you can work on that are the biggest pain, first off, because trying to chew this all off at one time can be a lot at times. So just try to find those 2 things that you're trying to work on first.
And the other thing I've learned as we've worked with irrigation, sometimes a first blend that we come with, we definitely inflict change, but maybe we're missing something. Maybe we missed the micros, or we didn't have quite enough potassium in to outcompete the sodium. So I think this year will really be a discovery period of getting our blends correct, and then we get to the next year, how do we build that into an economical program that is all encompassing to base rate fertility. So, that answer your question, Tim?
Yeah, appreciate that.
Any other questions here? If not, thank you guys. Appreciate it. Let's give them a round of applause.